So part of my job is leading the early phase clinical trial program in lymphoma research here at UCLH. And this is a really exciting part of my job because it allows me to potentially offer exciting new treatments to patients with lymphoma. This is often in the relapse setting where standard treatments have failed to get control of someone's lymphoma, or it's perhaps come back or relapsed after a number of lines of previous therapy. And the people looking after the patient, whether that's here at this hospital or another referring hospital, have decided that the current standard treatments no longer offer a chance of treating a patient's lymphoma successfully. Phase 1 trials are essentially the first step in testing whether a new anti-cancer treatment is effective and safe in patients. And the key to phase 1 trials is understanding that it's about really assessing the safety, the tolerability, the side effect profile, and optimal dosing of a new anti-cancer, specifically in my case, anti-lymphoma treatment. The other key difference to phase 3 trials to the patient is that these are quite intensive trials. These involve a lot of visits up to see us, to see either myself or one of my colleagues. This may be to look for whether there's any new side effects, whether that's symptoms that a patient may report, or through blood results, blood tests. So there's often quite a lot of blood tests required within a phase 1 trial to look for side effects, but also sometimes to measure how much drug is in a patient's bloodstream at any given point in time after a treatment has been given. So the frequency of visits is quite intense and does need to be taken into account when enrolling a patient in a trial. So one of the other key differences of phase 1 trials to, say, phase 2 and phase 3 trials is that these are trials of a limited number of patients, really looking for an early signal of safety and tolerability, and hopefully a signal of efficacy as well. So these trials are looking often to only recruit a small number of patients, often over quite a short timescale. So one of the challenges of the early phase trials is that it's a constantly shifting area where a trial may only be open for a short period of time in our centre until recruitment has been achieved. Another key difference to the phase 1 trials to phase 3 trials is, whereas a phase 3 trial may be open for a number of years and the site is free to recruit patients at the rate at which they see patients, phase 1 trials kind of go in a stop and start pattern, especially when a phase 1 trial is starting out and we're just establishing the safety of a new drug and the optimal dosing of a new drug. We talk about slot availability and cohort dosing quite a lot in phase 1 trials. Both of these concepts mean that slots are available for patients, often only in a short time window. And when I get referred a patient, I need to consider whether there may be a slot available for a patient, and that will be determined by the sponsor, and it will also be determined by where we are in what's called the cohort dosing. Often we'll enrol 3 patients at a low dose, stop recruitment at that point, watch those patients very closely over a period of time for the development of any side effects or toxicities, and if no side effects or toxicities emerge, the sponsor will then allow us to open up to the next cohort, often at a slightly higher dose. We then often allowed to recruit, for example, 3 more patients at the next dose, at which point recruitment again closes until those 3 patients have been assessed again for the safety and toxicity. This process continues until we see a side effect or a toxicity that tells us that perhaps the optimal dose of this treatment has been reached or sometimes even exceeded. So first-in-man studies are where a potential new anti-cancer treatment is used for the first time in human subjects. This may sound very daunting, but I'd be reassured that we only do first-in-man studies where there is an absolutely sound scientific rationale for testing a new drug in patients with relapsed or refractory lymphoma in whom there are no other treatment options. These drugs will have gone through rigorous testing in the lab to make sure that there's a good reason to believe it may work in the particular type of lymphoma we're looking at, and to get as good an understanding as we can about the potential side effects or toxicities experienced with this drug. The other really important thing to understand about a first-in-man study is that we start these treatments at an extremely low dose. The lab studies will have already established what we think may be the effective dose, but when we're first dosing these treatments in human subjects, we take this right back to a much lower dose, just in case there are side effects that are unforeseen from the lab studies.